విష్ణు అతను రాకపోతే పవన్ చెప్తే సార్ అక్కడ రావడం లేదు నాకు తెలియదు సురేష్కి తెలుస్తుంది అతను రమేష్ బాబు రాని
Can you display the schedule? आज का?
ఎస్ సార్ ఎస్ సార్ ఐ విల్
Second page.
Okay. Good morning, everyone. Our today's chief guest, Professor S.C. Lakhotia, distinguished Professor PHU and SERB Fellow, NIB Director Dr. G. Taru Sharma, Dr. Subir Majumdar, distinguished Professor NIAB, Dr. Hegde, Head Academics and Research, Dr. Redenna and Dr. Majumdar from HCU. Dr. Sharmistha from HCU, and distinguished people who have joined us from YouTube online mode, students, fellows, NIV staff, and my fellow colleagues. I welcome you all to this 11th Foundation Day of National Institute of Animal Biotechnology. Well, my first and foremost duty to thank the founders of this great institution who had a vision of imparting such a world class of research institute. Clearly, without their vision, constant support, and blessings, this institute would not have become what it is today. I also take this opportunity to especially thank to the faculty and staff who have been with this institute even since the inception and shaped the institute where it stands today. I'm also glad, very glad to see the active participation from all the departments of the institution during the almost 1 month long celebration of this foundation, in fact, Foundation Month. People have been very enthusiastically involved in various activities, be it walkathon, be it plantation drive, sports activities, and I don't have to mention what happened yesterday evening. It was a blockbuster performances by students. Probably this energy and enthusiasm of NIB people makes this campus more vibrant, and I'm sure in coming days and coming years, it will continue to grow. Above all, what could be better than having Professor Lakhotia with us on this day for delivering the Foundation Day lecture. Thank you very much, sir, for being us on this day and enlightening us with your experience and knowledge. Once again, I welcome you all to the 11th Foundation Day celebration, and thank you very much for being with us on this day. With this positive note, I would like to invite NIB Director Dr. G. Taru Sharma to brief the audience about NIB. Ma'am, please.
Thank you very much, Dr. Sanjay. Very good morning to one and all. We're the chief guest of this morning, Dr. Lakhotia. Thank you very much for being with us and having accepted our invitation to be here and giving the foundation day lecture. Before I address others, good wishes, best wishes for our foundation day to one and all, to my colleagues and also to students and fellows, all the staff of this institute. Dr. Sanjay has already detailed out that about our 11th foundation day, and it is such a good morning that we have previous directors, the founder director and the director who has shaped and made this institute to the level at which it is. And I'll fail in my duties if I do not mention the director in charge for the 6 months who has equally played an equally important role to take it ahead. I'm talking about Dr. Shivu. So with that, I just start. I thought I would be extempore, but then I thought Professor Lakhotia is visiting us for the first time, so it would be very apt if I take you around to the. It doesn't work. Sure. All right. So as I said, that this is our 11th foundation day. So it's no longer a decade old. So we started growing from 2011. This is one of the new generation institute, a brainchild of Dr. Maharaj K. Bhan, and he has planned and executed a couple of institutes of this country, and we are one amongst them. And all our planning was like that. It was he, Dr. Joan, Dr. A.K. Rawat, and that time Dr. Redana joined hands till it was all happening in the initial days. And after that, 2014 to 2018, we have had an interim campus. It was housed at Mayapur. And it was 11th of August 2018, with all the sincere efforts of everybody of this institute who were there at that time, and before that, under a very dynamic and able leadership of Dr. Subhi, who was the director when people entered to this campus. And that's how today we are celebrating from 25th May 2011 to 2020. All right, so this is a kind of vision and mission. It's very simple. Our motto is we have to take care of the animals, production and health for human welfare. This is troubling me. Right? Thank you. So these are our research focus areas. Precisely, we can put them under 6 subheads. As I was talking, we do both. Production biology group is as strong as infectious biology group, along with a very robust team of bioinformatics and nanotechnology people with us. So these are the key research areas, and we touch upon all these important pathogens, so viral, bacterial, and parasitic, and other delivery system adjuvancy work. This is the area in the reproductive biotechnology. Genomics, transgenics, and stem cell is also transgenic technology. I think it will not be hyperbolic if I say in the country not many people are working into it. It used to be the laboratory of Professor Kulani Shishakri in Institute of Science who was doing things, and he did generate a transgenic mouse model. It was good. However, it's not that vibrant as it should be. And then it was led by Dr. Majumdar, and we are looking for ahead with the newer plans to go on. This is the team bioinformatics is involved with hand-holding with majority of the scientists of this institute. And this is a nanobiology group with the diagnostics and nanodelivery. And the nutrition, nutrigenomics and enriching the animals with important nutrients. We have a couple of important flagship programs wherein zoonosis and One Health is a is one of our pride programs, wherein we have a big consortia. It includes medicos, veterinarians, basic biologists, and a lot of veterinary officers on field, and besides that, a wildlife institution. Another important flagship project we have is on livestock genomics, and this is again kind of jewel in the crown of the institute, wherein the institute could generate a HD chip, which is 7,88,496 steps into it. It was released by Honorable Minister last year on 13th August 2021. This is India's first and world's largest, because the other one has 77,496. So we are ahead by 11,000. This is the third flagship project which we have at the institute, which is also focusing on the zoonotic important pathogens, Leptospira, one bacterial and one viral. And this is what is our third flagship program. We have, there are a couple of good leads for both the pathogens. And there is a small platform which we could generate based on the electrochemical sensing method for JEB. This is the reproductive biotechnology team, which is trying hard somehow to increase the fertilization ability and address the oocyte aging. And we do a lot of work related to that and in parallel to that. So ultimately, the idea is that this is all we are doing in a mouse model and translating it to caprine model, a small ruminant, with an aim to take it ahead with a large ruminant. This is the picture which reflects that how we are doing in the extramural project. So we have 89 total, and 46 we have completed out of which, so we're doing good. And the total capital through these EMRs are good enough. This is over and above our basic core project funding. This is the picture of our students. Currently, there are 64. We are working towards CB. Earlier, the institute was a couple of faculties. We are still continuing having the registration through Manipal. And these are the papers per year. Professor Lakhotia and me this morning, we were talking about the H-index and the impact factor. I think I'll leave it on you, sir, when you speak and your opinion about it. However, as it's in vogue, so I do put that we are having the average impact factor per site is 4.94 for the last year 2021 and 22. This is a picture of IP and tech transfer. We have one patent at place and the other technology transfer which is in process, and in fact a couple of it has already happened. These are one for the clinical subclinical mastitis kit. We could already transfer to a company, and IP is already filed with that. And this particular company is also an incubator with us. Dr. Redana, this is our first incubator at our incubation center at NIAD. This is the second one, the group which works on brucellosis, pretty focused, and they have generated, they have a specific protein, BMP5, and they have developed LFAs and ELISA, the protein and the peptide, they are into it, and they are also, they have put a, like applied for the patent, and technology transfer, one is done, one is in process, and they are further going for the new planning for the new generation vaccine. Again, the aptamer base of ciprofloxacin antibiotic in milk. This one is a transfer detection kit, IP5, and they publish a paper also, Komal has already published. This is again from the same laboratory where they have the antibiotic sensitivity testing incubator, and this is to treat mastitis. As I was talking, this is an important disease at field level. So the group does, they have transfer technology. And this one we are looking for, and we are in a process to transfer it. So for the Toxoplasma gondii, very important parasitic infection, the team has generated a point of care diagnostics for that. They have generated this LFA, and this is, they have also done the validation. The output is appreciable, and they look forward for the transfer. These are a couple of new initiatives we are really looking for, especially on the delivery, and wherein important part is we have to do the human bypass. So protein is available to these animals, especially ruminants, and this is, it's a hand-holding one between 2 to 3 laboratories. So we address the problem, And this is a very important problem to address the reproductive inefficiency both in males as well as in females. However, our scientists have taken up in the males only. So these are the endocrine disruptors, the kind of feed, water, we really don't know, and it's a farmer doorstep problem. About then again the establishing of organic culture and the stem cell biology, making it more robust so we can really go further with the therapeutics and rush quickly through this. These are our, we do science, we also do some of Government of India flagship projects, Science Setu, social economic activities, and outreach program, Milan, which Dr. Subir and some of the scientists they plan, and we would continue again to that. And these are MOUs signed. We have signed a couple of others which are not here, but a good number. And these are a couple of major events which happened last year at NIAB. These are two important workshops and symposia we could conduct last year. And these are the major events put in one capsule. Besides, the vaccine testing facility was established. A lot of efforts were given by Dr. Hegde and his team, and they're continuing it very well, going ahead for accrediting this facility. These are awards. Our colleagues are doing very well into this also. And this is the major outcome. I think I'll not give more time. I'm going to cover to it. Thank you so much. I think Dr. Sanjay took 1 or 2 minutes extra. With that, I thank one and all. And I request Dr. Hegde to please welcome Professor Lakhotia, requesting Nilanjana to please assist doing that.
So, very good morning to one and all. And Professor, as we all know that Professor Lakhotia does not require any introduction. However, it is important to know that Professor Lakhotia is an Indian cytogeneticist and a professor at Banaras Hindu University. He is a distinguished professor at the Department of Zoology and INSA Senior Scientist. He is a Raja Ramana Fellow of Indian National Science Academy and the Indian Academy of Sciences and the National Academy of Sciences of India. In 1989, the CSIR, the Council of Scientific and Industrial Research, India, main scientific research organization, honored him with the Shanti Swarup Bhatnagar Prize for Science and Technology, which is one of the country's highest science honor, for his contribution in biological research. Not only this, Professor Lakhotia has many awards on his name, like INSA Young Scientist Medal, INSA SL Hora Medal, INSA Jawaharlal Nehru Birth Centenary Visiting Fellowship, UGC Career Award, UGC National Lecturership Award, UGC JC Bose Medal, and Professor Siena Rao Medal for Excellence in Science, and he is a member of several editorial boards and advisory committees. Currently, Professor Lakhotia is a distinguished professor and Sir-Distinguished Fellow at BHU Varanasi. The scientific interest of Professor Lakhotia includes cytogenetics, gene expression, cell and developmental biology. He conducted a groundbreaking studies on Drosophila fruit fly, on chromosomal architecture and replication, as well as the arrangement and functions of Drosophila melanogaster long non-coding RNase 93B, or DHSR Omega G locus. He has over 165 original research papers, review articles, and book chapters published. Professor Lakhotia was instrumental in the development of Banaras Hindu University's Department of Molecular and Human Genetics Department. With this, I welcome Professor Lakhotia to come on the stage and enlighten us.
Okay, good morning, everyone. It's a great honor for me to be here and to celebrate the foundation day of this institute. It's my first visit, and I must thank Dr. Sharma for letting me have this opportunity to be with you all, to share the excitement of foundation, and also share some of my ideas which about what we should do as a biologist. When I'm not going to give a typical research talk, but just to highlight something that many of us have been missing, and particularly for students, something that they may be aware or getting aware in recent years, but something that they have learned in a different way. And I'm going to explain that some of this learning we need to unlearn. And that process, how this institute's mandate and direction can move ahead, I will leave that to all of you to think about. But what I will do is just give you my current research interest. That's, as I was stated, the non-coding HSR omega gene in development and so on. Then we also started looking at the stress proteins, the molecular chaperones in cancer. And of course, I'm sorry. The other thing that we started looking at was Ayurvedic biology, that how the traditional formulations of Ayurveda really work. We all talk about Ayurveda in great ways, but then unfortunately, no mechanistic studies have ever been done as to what do they do if they do something, what's the cellular basis, what's the molecular events, and so on. And this was an area that started by Professor Valiathan, a cardiothoracic surgeon. He started this thing, and I joined in, and I really enjoyed working on this to understand how these formulations really work. Unfortunately, the Ayurvedic community itself has put itself in a shell. They don't want it, and I think we need to get out of that and learn so that we can really derive the benefits of this. That's the other thing. Of course, all my monologue is based 99.9% Drosophila, the little fruit fly. I'm sure many of the students would be familiar with this. And of course, beside this, I've also been interested for last 2-3 decades on higher education research assessments. I keep on writing on these issues about the policies, about journals, about ethics, and so on. So that's other area that I remain engaged with. Now, I'll just start with this, a very general thing. I'm sure each one is familiar with this. Miracle of life. I mean, the diversity. We, as a biology student, when I started learning biology, although like most students do, even in my own time, I took biology to be a medical doctor, but I was very poor student in school and couldn't make it. Then I took to biology, and then I decided that yes, biology is my area, which I really enjoy. And in that, one of the things that excited me, which continues to still excite, is the diversity, the precision and the diversity, how it is maintained. And so that's where the question was, how do we do this diversity? How do we maintain it? And that's what I'll try to explain in some ways, that how does it go on. One of the examples of miracle of biology is again like our body. This, of course, I'm sure everybody recognizes Michelangelo's famous sculpture, David, which is supposed to be one of the finest anatomy of the human body. The reason I put it here is that these bodies, the sculptures are created by somebody from outside, starting from an undifferentiated piece of stone or mud and so on, and then put them together, cut them from outside and chisel them and give the shape. The question is, how is human body made? Living body. Does it happen that way? I'm sure everybody knows that it doesn't happen that way. It starts with an egg and sperm and so on and so forth. And as a biologist, the question is, what happens during all these stages? How do they come about? How do they automatically keep taking these shapes? What are the questions? What are the ways? What are the mechanisms? And that's where the question is, how does it happen? And so in Miracle of Life, the question that we ask is, what makes biological system and process so precise and reproducible? Every time it goes on, barring some variations, which is what, of course, has given rise to all this evolution and so on. But then basically it goes on with great precision. And then how is order attained out of disorder? When we look at, I'm sure as a physicist, everybody knows that entropy is the basic thing. And to remove entropy, we need to put in energy and so on. So in biological system, again, should follow the same law of thermodynamics. Get to disorder, but then how to maintain order amidst this disorder? That's an important issue. And that's where we learn genetic information at the core of life processes. And that's where my interest, I should share this. In my master's degree on zoology and comparative neurology, I learned parasitology, and I enjoyed learning parasites of animals and so on and so forth. But in that process, I got interested in genetics. And so moved to a supervisor to learn genetics, and with the idea that I can go back to these animal parasites and see what things happen, but that has never happened. The Drosophila was so good that I have remained hooked for life with that little fly. And that's what I will share, what I've learned out of this. Now, that's where we learn in genetics that when we start from Mendel's law, that they established gene. Of course, Mendel called it factors, but then Morgan, they know what the gene term was given, and Morgan School with Drosophila laid the foundation of genetics as we know it today. Then, of course, 1942, Biddle and Tatum showed what one gene does, one gene, one enzyme. Because prior to that, gene was an unknown hypothetical entity. What it does, nobody knew how it functioned. All they knew was gene, and there's a phenotype. It mutates, phenotype changes. But then this was first time that some mechanism was provided that each gene produces a protein. Coding word was not known yet. It was just producing one. And then Avery et al. Showed DNA as genetic material. In between 51, 55, independent of the GLDG material, something that was established by pure cytology, histochemistry and cytochemistry, that there is a relationship between RNA and protein. Says that synthesize more protein, secretory proteins have more RNA. This was known. Why, how, that was not understood, but not even questioned at that time. And then, of course, Watson and Crick made this great thing in 1953 that we knew DNA double helix, and then things started, molecular biology started, and which many people may not know that the idea of genetic code was not given by any geneticist. George Gamow, an astrophysicist, a theoretical physicist in Russia, when he read Watson Crick's paper in Nature, immediately began thinking, and he wrote a letter to Crick that since there are 20 amino acids, and we know genes make protein, and DNA has only 4 bases, there must be a code language. This is remarkable. A physicist who had never learned biology, as a theoretical physicist, begins to think of this. And he predicted that the code must be triplet, because 3 raised to the power 4 gives you 64 codons, and you need only 20. And so, the idea was generated then, and that's what led, I'll come to that later on, the Crick's coming to central dogma and so on. And then, of course, Crick proposed the central dogma. And then Jacob Monod suggested a messenger RNA, that when they were working on the laxioperon, the beta-glucosidase synthesis, they talked about the term messenger RNA, which takes information from DNA to RNA, to protein. But then in 50s and 60s and the early 70s, a lot of cytological studies have gone on, biochemical studies on nuclear RNA and heterochromatin, the condensed chromatin, which was worked by cytologists, and biochemists worked on the RNA. But then, unfortunately, this information remained mostly suppressed, not really encouraged, because I'm sorry, this red color is not visible. If these lights can be turned off, it can be a little better, the front lights. That the in 1980 came the concept of junk DNA, selfish DNA, created primarily by Crick, that there's selfish DNA, and I'll come to this, that how this went wrong, how this concept was erroneous based on misinterpretation, misunderstanding of biological system. And this then led to suppression of this, all these papers, that there is a lot of nuclear RNA and heterochromatin transcribing that was completely ignored. And then, of course, 1900 onwards, 1990s, it should have, not 1900s, revival of interest in non-coding RNAs, and 2000 onwards, non-coding RNAs are now everywhere. If you look at any journal in biology today, any molecular biology journal, you will see every issue having papers and papers on non-coding RNAs. And so I'll just try to explain why this has happened. This is, of course, Crick's original drawing in 1956. When he thought, when Gamow suggested that there is a genetic code, then that led him to think about this. This is actually as he had typed and drawn himself. So that DNA makes RNA. He wondered, can DNA make direct protein? But RNA can make protein there. So this was question. And he put this RNA to DNA as an uncertain line, which Crick didn't believe for almost next 15, 16 years. Even in when reverse transcripts were discovered, he didn't believe it, that eukaryotes have it. And that's why the Nobel Prize for reverse transcriptase waited till Crick said, yes, it can exist. This is something interesting. Crick was such a dominating figure. And then, of course, RNA can make RNA, DNA can make DNA. But what he said was, this cannot happen. Once information has gone out of DNA, it cannot come back to nucleic acids. And this is the most important point to remember the central dogma. Central dogma is only talks about the path of information transfer. It doesn't say anything which is more important, which is less important. But unfortunately, we kind of misinterpreted this. And then, of course, this became his final updated central dogma. With DNA can replicate, it can transcribe, it can reverse transcribe RNA, and RNA can make protein. So some of the errors from this initial drawing were lost, and this error was confirmed. That's what everybody has learned about it. Now, one important thing that happened in biology, cell biology, cytology, genetics, and so on was the C-Bell paradox. C-Bell is the haploid amount of DNA of any genome. And that turned out to be that there's enormous variation in the amount of DNA that we have. For example, we will be surprised, an amoeba, single cell animal, some species have 10 times more DNA than humans have. Some frogs have 100 times more DNA than what we have. Do they have more genetic information? Do they make many more kinds of proteins? This was not found to be the case, and that's become paradox. That why is the DNA content in the genome not paralleling the biological complexity? Humans, of course, in their anthropocentric view, think that we are the best and most highly evolved. So it is insulting to see that a lowly amoeba has more DNA than us. And that becomes central C-value paradox, that why should it happen? And that is what resulted in this idea of junk and selfish DNA. So that's what it was. And because it was believed that genes, because in central dogma, we learned that genes make RNA, RNA makes protein. And Watson, in his Molecular Biology of Gene 1965 edition, I believe, wrote this simplistic statement: genes make RNA, makes protein. And therefore, an RNA must make a protein. If an RNA or DNA doesn't make protein, what it does? That was C-Bell paradox, why it is there. But the fact is, it was there. So how do we explain this DNA? So much of extra DNA, even in humans. The problem, when in 2000, when the human genome was completed, we learned that only 2% of our DNA makes proteins. What 98% is doing, we didn't know. And that's what, of course, understanding gave rise to the idea of that RNA can also have an important function. So central dogma proposed that Crick suggested that genetic information flows from nucleic acids to protein, but not in reverse direction, and that proteins find, as I said before. But then, because there's a lot of DNA that doesn't make protein, they didn't know its function, and so they called it junk or selfish. Junk term has a problem. because junk in English means something that is of not use today, but was of use in past time. So again, the question begs, if it didn't have any function today, what function did it have in earlier times? We also don't know that. So better was selfish. Okay, it's there, we can't help it. It has invaded our genome and will remain, we can't do anything about it. It doesn't do any harm, it doesn't do any good. And this resulted in any study talking about DNA that doesn't make protein, useless. And I know that, I mean, as I say, I started working on this non-coding RNA way back in 1970s. 1982, we established this. Fortunately for us in India, I could get funded for working on this, so-called selfish by that time. But my competitors in US, Heidelberg, MIT, they couldn't get funded for working on this gene. They just started working on the gene following our discoveries, but they couldn't get funded because wastage of money on selfish DNA. I'm glad that in India we could continue this, and I hope we continue to do that. And I would mention that I got Bhatnagar Prize for this non-coding DNA. That gene was the thing on which I worked. So I'm very happy that we have. I do not know whether the people who sat on judgment in my Bhatnagar Prize understood what they are doing, but they thought, okay, something must be there. That's all. So, but that's what it is. So the problem, why did this happen? Why an intelligent person like Crick missed this basic point? And I think that's where biology understanding is necessary. Crick, molecular biology, reductionist approach of physics that happened in, you see, if you look at biology history, we started describing animals and plants and so on, and we looked in totality. But then when molecular biology started, it brought with us the reductionist approach of physicists and chemists and mathematicians, that everything must be broken down to the lowest entity and that should be common. And so a gene makes RNA, RNA makes protein, and protein makes protein. Therefore, proteins are only important. And that's what Crick went on. And but then evolutionary biologists, as I will quote later on, always talked about that what evolves is not proteins. What evolves is the regulation. And I just give a very basic example. All of us have same face, same two eyes, nose and ear and so on and so forth. But shapes are different. Is it because their proteins are different? Proteins are same. What is different is how, where they are expressed, in what shape an organ cell divides, what direction it divides, how many times it divides. That decides whether a nose is parrot-like or sharp nose or a flat nose or a wide nose, whatever it is. So this is where evolution is already talked in 1960s. It's a regulation. Even Britton Davidson, who talked about repeated DNA, why there's so much of DNA, had stated very clearly that the extra DNA that many organs have has a role in regulation. But unfortunately, this issue was missed because of disconnect between cytologists, evolution biologists, and molecular biologists. Molecular biologists thought that they are old-fashioned. Let's not worry about them. And the cytologists thought, okay, they are young, new people, they don't know what it is, and therefore don't talk. That disconnect is where we get into problem. We need to learn it. So I just go back to some of the things that when I said that even in 40s and 50s, people had talked about protein and RNA. One simple study was that they looked at the classical biochemistry ways, how much protein tissue has and how much RNA it has. They didn't have RNA sequencing and all those fancy things that we can do today. They didn't have direct spectrophotometer which could measure RNA content. It had to be done in a very hard way. But then they found that there's a correlation between the quantity of RNA in the pellet and the rate of protein synthesis in the tissue. If they compare 3 different tissues, pancreas, liver, and kidney from rats or mouse, and found that pancreas, which has much more protein synthesis because of secretory activity, has more RNA. Liver, when it is active, has more, but kidney, which has much less protein secretion, has much less RNA. And therefore, that was the basic idea, to say that RNA and protein have a relationship. Which gives rise to what? That was not clear. But this is what Gamow made use of this information to suggest that between gene and protein, RNA must exist. That was the basic idea. But in 1960s, there were lots of studies on RNA. And there are many studies that have shown that there are many heterogeneous nuclear RNAs, diverse sizes of RNA, which are produced in nucleus but never go to cytoplasm. And by that time, it was established in 1960 that protein synthesis takes place in cytoplasm. So now if an RNA doesn't go to cytoplasm, it is a protein. What it does. This was a big question in 1960. There are lots of papers on this. That this enormous nuclear RNA which doesn't see the face of cytoplasm and what it does. That was a big question. They didn't know. And then there was a series of studies that showed that there is a lot of chromosomal RNA associated with chromosomes, which again they didn't know what it does. And they talked about that the repetitive DNA also transcribes but then all these remain unknown, unexplored, thanks to selfish DNA, thanks to junk DNA. But I think these studies are prelude to understand thinking about non-coding RNA, that there must be some RNA which does something which we do not know if we take the protein as the major component. But unfortunately, this direction remained almost inhibited for 2 decades. It was not actually supported. Some labs went on working on this, and there were some ideas. And 2000, when the genomes were known, human genome and other genomes were known, and by that time, the RNA interference phenomena were discovered. And so suddenly, people realized that there is something that can be done about it. And so this is what I said, that the problem was people took central dogma as a dogma. By the way, in biology and science, we don't talk of dogma. But unfortunately, molecular biology is based on this dogma, which is wrong. And that's where we are now trying to relearn the whole process. And so this became a textbook belief among molecular biology that DNA makes RNA, makes protein. And anything that doesn't do this, that DNA is of no use and so on. But
what was the experiment where we found that this selfish DNA is there?
I'll come to that. I'll come to that. Well, selfish DNA was a concept.
Yeah.
That DNA, because it doesn't make, it makes RNA, but it doesn't make protein. The only RNA that doesn't make protein were considered to be functional were ribosomal RNA and tRNAs and snRNA, small nuclear RNA, because they were related with protein synthesis in some way, so they were valid. But others whose function they didn't know, they didn't realize, they didn't want to explore, simply put them in a basket of junk DNA or selfish DNA. But so it was not, see, nothing was discovered as a selfish DNA. Only it was a negative description that because we don't know their function and they vary so much in related species. For example, even 2 related species of frogs which look alike can have the genomic DNA differ by a factor of 10. So the question was, there is no logic in this. Unfortunately, what we must understand in biology, sum of parts do not make the whole. It's not equal to whole. Unlike in mathematics, 2 + 2 is always 4. But in biology, you take nucleus, you take cytoplasm, mix them together. Humpty Dumpty, I'm sure you remember they couldn't be put together again. And that's what biology is. And that's where we need to go beyond reductionist approach. That's the point I'm trying to emphasize. So this as I said, block studies on nuclear and non-coding RNA for several decades. Now, how I got interested in this? My interest started way back in 1960s. As I was doing my master's and then PhDs, and I was reading literature, I got interested in heterochromatic. I was working on dosage compensation, where the X goes into Drosophila becomes hyperactive to achieve dosage compensation. That was one of my early contributions to biological system. But then by doing this, looking at chromatin organization, heterochromatin, condensed chromatin, and there were reports coming about repeated DNA being present in heterochromatin, and result that they were sometimes transcribed. So that was one of the reasons why I got interested, that heterochromatin was an enigmatic but a universal eukaryotic genome. Every species had this. Some species may have 80 to 90% of chromatin as condensed chromatin all the time. Some may have only 2 to 3%. It varies, but it is always there. The other important point that was there was heterochromatin, and especially the repeated and satellite DNA that are associated with it, evolved very rapidly. Between 2 related species, you look at the repeated DNA content, it's very different. So how does it evolve so rapidly? And if it evolves so rapidly, can it have a function? But then evolution biology suggested that this is an important function that evolves rapidly, because then it ensures repetitive isolation. Two related populations, if they differ in the repeated sequences, they cannot have good meiosis. They cannot have chromosome pairing during meiotic process, and therefore they are separated. And this was something evolution biologists in 60s and 70s established, that this is important for evolution. And then it was also known that several classical heterochromatic regions in Drosophila found to be transcriptionally active. They are typical heterochromatin, devoid of typical Mendelian genes, but still they transcribe. And the gene 93D gene that I got interested way back in 1970, we found it was not coding. And that's what had sustained my interest, and I've gone along with this. So for example, this was classical example of Y chromosome Drosophila, which is completely heterochromatin. I'm sure students will remember, others may remember, that you can have just X chromosome carrying individual, no Y chromosome. It's perfectly normal, viable. Only problem is sterile. And that's where it was shown that Y chromosome in spermatocytes completely opens up, transcriptionally active. And in 1960, they have shown that the RNA that they made by these regions of Y chromosome can be 1300, 1400 kilobase long. Huge RNA, nobody understood what it is. But it was shown that, yes, they transcribe. And this caught my interest that, okay, heterochromatin cannot be meaningless if it is necessary. And it was, of course, shown that if any of these structures that are there, if you have a deletion of that, spermatid doesn't differentiate. Up to spermatid stage, it goes all right, but then it doesn't differentiate. And that's why we know that the heterochromatic region is more important. For example, similarly, Lalji Singh later on in 90s or 80s could show that the W chromatin in snakes opens up in oocytes. If that doesn't open up, oocyte doesn't grow well. The equivalent of Y chromosome, the W chromosome in snakes, because they have the reverse sex determination system. So this was something that happens. And now we know that even in humans, Y chromosome does transcribe during spermatogenesis, and this is critical. So this was one interest that I had in heterochromatin in my historical learning phase, that I got interested in this. The other one that I myself showed that typical heterochromatin in Drosophila is transcriptionally active. It contains ribonucleoprotein particles and is active interstitial uridine incorporated, that means RNA synthesis. And therefore, I never believed in cell-free DNA. I was always convinced that this is a wrong concept and we should go beyond it. Then the 93D locus, which, as I said, I got interested in 1970 during my PhD study, that this gene was singularly induced by brief amide treatment of polyethylene chromosomes. And I will not go in details, but this was that everything else RNA is inhibited except that this gene was transcribing very heavily. And I got interested in this gene, and I thought this will, way back in 1970, that this will be a good way to understand what genes do. At that time, you know, that genes become active, they make RNA, that they make protein. This thing was not clearly demonstrated in eukaryotes. It was understood, but a clear example didn't exist that only you know this gene and you know when it is transcribing, you can study this RNA, you can look. So that's what I thought I'll do when I start my own career, which of course took a long time because I didn't have enough money to do many things. But what we could show in 1982, see, it's a 12-year gap, that from this question that I started asking in 1980, I could get answer only in 1982. One thing we would show that every species of Drosophila that we examine has this amide-inducible locus, which is also induced by heat shock. See, the Drosophila has this advantage of polyethylene chromosomes where you can actually see under microscope a gene becoming active. When it pops out, it swells up, chromatin opens up. And then we could also show that when I activate the single gene, I don't see any of the heat shock proteins being synthesized. Now, I should tell you briefly, to do this gel electrophoresis, why it took 12 years was, I didn't have money to buy a slab gel electrophoresis apparatus. I didn't have money to buy a gel drying apparatus. So one of my students actually fabricated with his own hands a vertical slab gel apparatus, a gel drying apparatus. And then with great excitement, what we found was it doesn't make protein. A great disappointment in 1980, just 2 years back, Crick and Ono and so on say this is all useless. And we discovered the gene which we thought a great expectation to be non-coding. I didn't give up. I decided to continue, and that's what I followed. And I think I can have one more life to understand what the gene does. That's the excitement that it carries on. And of course, following our studies, these labs in Heidelberg and MIT, USA, cloned this gene. One of these lab, Heidelberg lab, you know how did it clone? It cut out this part of the chromosome and then cloned it by micro digestion on this slide. Under microscope, you cut out this region, take this in the DNA isolation drop, extract the DNA, do phenolchlorophene extraction on a drop on the microscope slide, take the precipitated DNA, which you can't see, and another drop, add restriction enzymes, digest it, add vector, ligase it, and then transform, and it worked. And they cloned it, sequenced it, and they confirmed it doesn't have open reading frame. It's non-coding. It evolves very, very rapidly in different species. Fortunately for me, but unfortunately for science, their funding stopped with this. That it's non-coding, selfish DNA, why waste money? And remember, a lab MIT, Madhu Pandu was a professor, was a fellow of US National Academy, who was chairing one of the task force in NIH. She would keep on writing to me, show some phenotypes so I can get funding for it. We don't get funding. I mean, we were in a regular touch, but that's what it was. And so that's where I argued, is it really junk? It's not junk. Because evolution biology in 1960 clearly believed, as I said, evolution is regulation, then the protein coding genes. The reductionist approach is what led to this idea of selfish and junk DNA. And so I'm glad that the concept of selfish and junk DNA itself is becoming junk now. It's a thing of past. We don't believe it anymore. I know many students would have learned it in the textbook and in the classroom, that this is what it is, but please unlearn this. This was a historical concept, which was there for some region, valid region. It did see the selfish DNA concept, the central dogma molecular biology did promote a normal development in molecular biology itself, gene expression, all the regulation, and so on, because of that. But then it did not let other side to grow as well, which is now growing up very well. Now, one of the two things that caught attention of non-coding DNA was phenomenon of X inactivation in humans and mammals, because it affected humans, therefore it got attraction, that inactive X chromosome is coated with a non-coding RNA, XIST, X inactive specific transcript. It was shown in 1992 that there's a huge 17 to 20 kilobase long RNA, which has to coat one of the two X chromosomes to make it inactive. And so here was a very clear phenotype and a very clear demonstration that there's a non-coding RNA, which is critical. If this is not functional, female can't develop properly. No female. Similarly, in Drosophila, Drosophilus companies that operates, but in these two groups, they operate in different ways. This is where I had contributed in the initial study that it's by hyperactivation. That the single X chromosome is transcription more active than each X chromosome in the female, and therefore it produces as much product as the two X chromosomes in females do. In mammals, it's the reverse. One of the X is inactivated, so each sex somatic cell in male and female mammals have only one X chromosome active, and therefore there's doses composition. But in Drosophila, RNA on X, two non-coding transcripts paint the entire X chromosome. They remain bound, and only when that happens, this X chromosome can become hyperactive. So here, where non-coding RNA that decided chromatin structure, and that's what we know in epigenetic regulation, that when this RNA binds to what all kinds of histone modifications and DNA methylation and so on process goes on, how does it happen? And same thing happens in this, that how the acetylation of specific histones happens because of this RNA being there. So this was one clear evidence in 1992 that non-coding RNAs are critical for very important functions. And then, of course, the RNA interference, which was discovered again accidentally, that there is a small RNAs can, as a biotechnological approach in plants, that if you express it too much, the whole thing goes silent. And RNA interference was discovered. And that led to a lot of biotechnological applications that, okay, we have microRNAs in humans as well. And if we can block a microRNA through some gene, we can regulate specific expression. That led to a lot of information. And in 2002, science declared small RNA as the molecule of the year. that here this is a very important RNA. And so then the interest in non-coding RNA revived again, because by this time the genome had also shown that 98% DNA is different. Between humans and chimpanzees, the coding sequences are nearly identical, but non-coding sequences are enormously different. And so more and more realization that non-coding DNA, non-coding RNAs are critical for function. Now, I'll just tell you about 93. What we could do was, it is, that it is developmentally active, stress-inducing, singular induced by amides, essential for survival. I mean, between 1982 to 2000, we published large number of papers describing this phenomenon. Only in 2000, we learned what it does. And today, of course, we know that this gene, a very long gene, nearly 20 kilobase long gene, makes 7 different transcripts using 2 different transcription start points, 4 termination points, and an intron which can be differentially spliced. And the nuclear RNAs are large, they carry repeat sequences, and they organize these small speckles in nucleoplasts, not on chromatin, which we named as omega speckle in 2000, they remain bound with this RNA, non-coding RNA, these 3 large non-coding RNAs, and a class of RNA binding protein, the heterogeneous nuclear RNA binding protein. They remain associated. And what we could show was that these HNRNPs and this RNA absolutely co-localize. They are present on the gene itself also where they are there. And this is what we call as omega speckles. And this now we have seen in every Drosophila species this happens. The gene is conserved, but sequence-wise, it varies enormously. The same function despite different sequences. And the question is, why should this happen? The small differences sequence in the RNA allow them very precise modulations. When a gene and when it will be activated and which HRNP will be released and so on, that's the mechanism that it can operate. So now, of course, we have a whole variety of non-coding RNAs. There are diverse bases of classification. I will come to some terms next week, but more common classification is based on their size. Short or small non-coding RNAs or long non-coding RNAs. Simply, less than 200 nucleotides is small, more than 200 is large. This is a very empirical classification. It doesn't tell you anything function, but it's still operative because we don't have any good way of classification. And why we don't have? Because there are so many different kinds of functions. You just can't put in one classification as such. The long non-coding RNAs, LNC RNAs are called enormous numbers, diversity and rows. And numbers are increasing every day. Every day you see papers talking about different non-coding RNA discovered bioinformatically or experimentally. And this has become a very interesting area to discover and to catalog. But then I think we need to go beyond stamp collection. We need to understand what these are. And these are different terms like sense and antisense, non-coding, long non-coding RNA, intronic RNA, promoter RNA, bidirectional RNA that you make in both orientations. Then we have intergenic LNC RNAs. Then mRNA like non-coding RNA, because some RNAs which are non-coding also have called open reading frame. So they can function as protein coding sequences as well as non-coding RNA. For example, the HSR omega, we now know that it makes a 27 amino acid peptide, and which we have established that it is made in the cells. But what function it does, we are still trying to understand whether there is a role in translation process or in the RNA life cycle or protein turnover. That's something that happens. And then we have architectural RNA. This I'll come to later on, that these RNAs provide scaffold for many of the structure that we have in the cells. And that's what has been called as architectural RNA. Then there are epigenetic link RNA that, like the ZIST and ROX, they provide the structure information, chromatin organization. Then there are translation long non-coding RNA, which are roles in translation regulation. Then we have circular RNA derived by back splicing of a typical mRNA. So that the product is covalently linked, it makes a circular RNA. And these are again becoming enormous variety. Initially it was okay, 1 or 2 were known, but now this is again a whole new class of circular RNA that are becoming known now. And we wrote recently a book chapter which one can see for all the details. And then, of course, I should say that way back in 1996, 95, I gave a lecture at a symposium called Trendies. This symposium, series of symposiums were organized by Professor Ram Sharma of IISc Bangalore. And the idea was not to present your work, but to predict what is likely to be useful in next 10 years or so. That was his theme. So I was invited by Dr. Hasnain at NII in 1995 meeting. And there I talked about that non-coding RNAs are going to be trendy. At that time, they were still under cloud, that are they junk or they are selfish, but that's what I wrote. And then I was asked by Indian Journal of Biochemistry and Biophysics editor that why do you write a review on this? So that's what I wrote. And one of the things that I wrote there was non-coding transcripts are no longer mere curiosities or vagaries of biological diversity. These seem to establish themselves as a distinct, at least I thought that they have established, but most of the people still didn't believe that they have any function, with very important function. Understanding the signal of such genes has been thwarted by common selfish genetic element applied to them. And with RNA being the first living molecule, it is but to be expected that even today biological systems continue to utilize this versatile molecule. Remember, origin of life, if it could depend on RNA, sustenance of life cannot be without RNA. RNA cannot be just a mediator of information transfer. It has to have fundamental functions. And that's another editor I wrote, followers of normal biology need to become less dogmatic about it, because biological systems are very diverse. They are just not follow the reference laws as such. And then we suggested that low non-coding RNAs can also function as hubs in cells to regulate a whole variety of systems. This was primarily based on our own studies on the HSR omega gene, the kind of interaction that we found. And so this is what we suggested for long non-coding RNA, that this single RNA, which is very sensitive to signals from outside and within, can have multiple roles to play because of interaction with variety of proteins. HNRP, they can do chromatin organization, they can RNA stability, splicing, trafficking, it can interact with nuclear matrix proteins, it can interact with molecular chaperones, it can interact with the cell death pathway gene. This is what we could show with using genetic and molecular data, that here these interactions are real, and then we built up this network that this RNA can decide whether the cell will survive or will die. And I think this is a prototype which can apply to many, many long non-coding RNA that to function. And that's where we suggest that these can be really hubs in the cell function. And what we now know is that what all things long non-coding, short and long non-coding RNAs can do is that they can make hairpin loop, they can be microRNA, which can regulate transcription, translation. They can, then some non-coding RNA can become sponges for them to regulate microRNA. You see, now you have two layers. One, microRNA regulating other mRNAs, other RNAs regulating microRNAs by acting as a sponge, so that they are all taken away, withdrawn, and can be released when they want. And similarly, the long non-coding RNA can form scaffolds, can form sponge, can do many variety of things. And in between, you get circular RNAs, which can do many other things. So there are a whole variety of things that RNA can do, which we are now beginning to understand. And of course, one thing as a biologist, we need to understand. Cell is an extremely crowded space. There is no free space in cell, not one single Armstrong area left blank. Something or the other is there. And so one can always ask this question, how do cells do what they do? How do molecules find their quick targets? We have learned of all gene regulation that comes a hormone and immediately a specific group of genes is activated. One has to really look at spatially that how that signal from membrane travels specifically and identify specific 6 or 8 base pair sequence on the promoter, and why only certain genes. For example, in steroid hormones, there are hundreds of genes that have the steroid hormone response elements. Why in a given cell type only some promoters respond, others do not? And when you begin to think in this crowded space, it really becomes mind-boggling that how does it happen with precision? How out of this complete disorder, we get order. And this is where RNA plays a very important role in that process. And that liquid phase separation, a physical event that was known to physicists for long, but historically, I should say, way back in 1898, not 1998, 1898, one of the psychologists has stated that cell is not a solution, it's an emulsion, where molecules are floating, and that's what he didn't know phase separation, E.B. Wilson, but he had predicted that something, and of course, they didn't have fancy microscopes and tools to them. They looked at with very ordinary microscopes that I'm sure most of us would have used in schools, typical monocular microscope. That's what they had at their fancy equipment, and they made that discovery. But now we know this liquid phase separation, that this is a very important process. Everywhere in the cell, we see these LLPS, membraneless organelles. Nucleolus is one of the largest membraneless organelles. Ribosomes, there's no membrane, but still they've maintained their entity. And we know all this process happened because of liquid phase separation. And this is what happens. In almost all of them, RNA lies at the base. RNA provides a scaffold for proteins to associate with it. And once it gets a critical shape and critical structure, it immediately gets phase separated. And that phase separated entity keeps the molecules in one place, but can also allow their release as and when required. And this is what is becoming a very important process now to think about in a cell. And we have a whole variety of organelles in the cell which are membraneless. See, Golgi apparatus has a membrane, but centriole has no membrane, but still it maintains its organization. Ribosomes have no membrane. There are a variety of nuclear speckles. I'll show you some examples. There are no membrane, but they maintain their integrity, and they become critical for cell function. For example, this omega speckle that we talked about, there are in mammalian cells, human cells, there are two other kinds of speckles known. One is the splicing speckles, which have an RNA, non-coding RNA, MALAT1 as its base, and then proteins like SA35. And when we look at the organization of the protein, what we see is RNA is on the outside. Similarly, paraspeckles, they have a protein, HNRNP, NONO, and a NE2 non-coding RNA. RNA is on the outside, protein is in the center. Now, remember, all these proteins are proteins that have low complexity. Either they have domains of low complexity, or they themselves are, the whole protein is low complexity. And that's where they can interact with a variety of other molecules, including RNA. And that's what allows them to be get involved in phase separation event. The omega speckle that we talked about, we can look at the RNA. This is by conventional confocal microscopy. If we do super resolution, we can see RNA and protein make a kind of meshwork. And now we are taking up studies because we know at least 10 different proteins are present in this. So we are planning to look at each protein where it is in this omega speckle, how they're organized, which part of the RNA is where. I think we now have the tools to do that, to look at the actual anatomy of omega speckles. But what I'm trying to illustrate is that these organizations happen at a very rapid pace. And when they happen, they impact total cellular activity. Even you see P bodies, where how the RNA turns over, they get into a particular cell structure, P body, which is again membraneless. That is because of this kind of RNA protein interaction. The human diseases like Huntington's or Alzheimer's or Parkinson's, they have these aggregates, again membraneless. And again, in these aggregates, some RNA seems to be at the base of it to give that kind of structure. And so this is where it's necessary that we keep on examining them. For example, in Drosophila, what we had seen was that these omega speckles are distributed all over. Within 5 minutes of heat shock, these proteins disappear, these omega speckles disappear, and the whole proteins are at the gene locus. Now, this is a very extremely fast sensitive way. We have looked at time-lapse imaging of this, that how do the proteins disappear from these speckles, how do they move to the locus, how does this RNA happen? And see, this is what is important, that these RNAs can provide very directional movement because of LLPS and because of other interactions that can happen. It looks typical example of order getting out of disorder, because these speckles appear to be distributed all over. Why they are distributed all over? You know, like these are supply depots. proteins and RNAs are there. A neighboring gene needs activity, needs the proteins. The omega speckle disappears, proteins are released, and they can quickly find their destination. This is one way that they can work on very fast way. And so this has become very important realization today, a very excitement in biology today that the LLPS, yes. No, they are these ones don't have lipids. They are only proteins and RNP, ribonucleoproteins. Yes, lipoproteins can also be emulsified, and thereby proteins can be trapped and lipids can be trapped. See, typically with again, unfortunately, what we think of always in terms of proteins. Yes. Well, no, they're not transported through lipoprotein. They're transported directly. See, these proteins can be soluble in the nucleoplasm as well, and therefore they can diffuse. This is what we have done using. No, RNA is not playing a role in transport. RNA is playing a role in keeping them at one place. Some signal comes, the proteins can get, for example, pyrylated. And once they are pyrylated, ribose, NAD activity, then they cannot bind with RNA, so they get out. There's a very quick process. And when they get out, RNA get degraded, proteins are released, they diffuse out, and because they're in such large quantity, they find a target, stick there, do their function. So we have done this whole dynamics of how these proteins diffuse out during the process. You see, even when they're concentrated, proteins are not jailed here. They can move out across this. And because this is what allows randomness, so that as soon as the things are better, the proteins can find the targets and they can function there. So now I also wrote a small, yes. Well, in the random in the sense that they are close to chromatin, that's all. That they are not specific genes where they are. No. You see, the Drosophila has only 4 centromeres and 8 telomeres. That's all. So, and of course, in Drosophila, centromere will be at one end, and telomere will be at the other end. I mean, in 3 dimension, if you look at it, we know that. That total 3-dimensional structure of chromosomes is also known how they are organized. So this is not with telomeres and centromeres. And of course, different nuclei will have at different developmental stages, will have more or fewer number of them. That also varies with the cell function. So this note that I wrote was industrial gene and beanbag genetics. This is where biotechnology was bothering me a little. Let me be honest. The way we were teaching biotechnology in our country, I've been really worried about it. And I actually wrote an article in Current Science that they know neither technology nor biology. Because all that we teach, I'm sure students will agree with me, PCR or gel suppression or restriction digestion. Why, how, and so on, the biological context was not explained to them. And this was happening that, okay, everybody was trying to make use of this to apply to develop some application and some diagnosis. And so then I wrote that industrial gene, that's a biotechnological applications of this, and beanbag genetics. This was a term that was a hot point of discussion in great geneticists like Halden and Muller and so on, because what beanbag genetics means is as if genes are a bag of beans. Each bean being one gene, you pick up this and you get this function. You remove this, you lose this function. And the argument that was being made was that our genome is not a bean bag. It's a complex organization. And so one of the things that I wrote in that was, intense research driven by biotechnology has, of course, remarkably improved our understanding of basic life processes. Yet, we still do not know the full protein coding component of our genome. This was in 2000. Even today, we don't know the full protein coding component. We keep on discovering new proteins and their genes and regulations, let alone the near complete ignorance of bulk of the known coding components, much of which is obviously involved in regulatory networks. Therefore, to think of manipulating any genome to our advantage with the little knowledge that we have even today is premature and frightening. And I think it's applicable even today. And that's where I thought I should sensitize that we need to look in a holistic manner. And particularly, I also quoted a statement by great evolution biologist Ernst Mayr, who was written way back in 1970, that who was worried about considering structural protein coding genes only to be of significance. So already he was worried about the in anticipation of selfish DNA concept, because people were talking about it. So he did say they will come when much of the population genes will have to be rewritten in terms of interaction between regulator and structural genes. And this is where the non-coding component and the coding component becomes important, and we need to understand that. And of course, today we now know that there are lots of diseases in humans and animals which are based on non-coding, which have some relation with non-coding RNAs. There are large number of these small and long non-coding are being associated. I'm sure every day if you look at, you see new paper coming out. And they have been identified as biomarkers and therapeutic targets. That look at this a biomarker, this microRNA or long non-coding goes high up in something, and therefore it can be used as a biomarker to diagnose that gene, because these RNAs happen to be serum as well, although they are being made in some cancer tissue, but these RNAs happen to be in serum, and therefore they can be good marker, non-invasive biomarker, very good. But what we need to understand is, what of these non-coding RNA do in a normal cell? Only when we understand that, we can really use them for therapeutic approach. We simply say, okay, if this RNA is more, therefore I kill this RNA and disease will be gone. I'm sure things will be problematic, because that RNA may be involved in hundreds of different functions in different cells, normal cells. And that's why it's necessary to understand these things in a better way. And that's where some of the things, one of the articles that I wrote in Nucleus and Indian Journal, by the way, I should, which you mentioned, I have taken as my personal policy for last 50 years to publish half of my research in Indian journals. Only thing my consideration is that they must have good reviewing policy. If a journal publishes because it comes from Lakhotia, I don't go back to that journal. Once I've withdrawn my paper because it was not properly reviewed. But I mean, to the young people and to colleagues and so on, please do consider good Indian journals published. And then only when we publish good articles in them, world will know about it. Otherwise, we remain in the vicious circle. Like as I was mentioning, I wrote an editorial in Current Science 2-3 years back. Title was, "Why have Indian journals not made a mark? Enemy is us." We are our own enemies. Because as a senior scientist, a paper comes from Indian journal for reviewing to me, forget it. It comes from some Timbuktu journal, rush to review it. Unfortunately, yes, this happens. You go with a paper in Indian journal, the selection committee chairman and the others will say, oh, useless, don't worry about it. We need to change that. And I think the young generation which has to take this challenge and tell the people who sit on the judgment chair bluntly, look at my work, not the journal. And this is, I think, what we need. And so I keep on writing in Indian journals. Even today, one article has come out in Current Science on how do we combat predatory journals. What thing we can do, what countries should do, what administrators should do, that we stop this menace of predatory journals. And 2 days back, somebody, after reading this, wrote to me, what do you think of predatory patents? And that's what I learned, that you can buy a patent. And this company was offering 10,000 rupees. They have already listed hundreds of patents. They are ready for filing. All that you need to pay is 10,000 or 12,000 rupees. Unfortunate situation here. And I think an institute which has applied work, everybody must be conscious about this kind of situation. That we don't get into this rat race of claiming patents, which we have not done, or publishing papers which are not worthy of being cited, not worthy of being read. So that's where I have been writing this. And okay, so what I had written there was about the biological systems are not created by design. This, of course, not my own idea. This was something that was stated by Franco Jacob of Jacob and Mono, who had worked that paper, that action of natural selection has often been compared to that of an engineer. Natural selection is not an engineer. It's just a tinkerer. That's what he said. That whatever is available in the genome, it tinkers with it and creates a new genome and new organs. It's not an engineer who had designed that, okay, I want this gene, put them together and I will get a new organism. No, that's, of course, today we are talking of artificial and new biological system that we can create, but we can create only up to limited level. What happens in natural selection perfection is something that we need to worry about. The C-value paradox, again, I explained that if a species can tolerate enormous selfish DNA, so-called selfish, for example, transposable elements, people talk that they come in. But what we are not realizing today, and now we are beginning to that, transposable elements are not just sitting there to transport themselves. They have been repurposed by our genome for our own process. In fact, the two tissues in which transposable elements are most active are germ cells and our brain. If they are purely selfish, why should they be allowed to transcribe so much? It needs to be examined whether their transposition generates some desired genetic rearrangements or gene expression patterns, which lies at the base of brain function. That this is something that we need to look at. And therefore, simply ignoring them as they are selfish, we are simply ignoring many basic questions. And so conventional reduction approach cannot explain the variations and of course the diversity in life system that we have. And biological systems are products of chance and necessity. In fact, Monod has a book by the title Chance and Necessity. And I think we should read that to understand philosophy of biological organization, biological systems. And there have been some other articles that I've written. So, and I think non-coding RNA heterochromatin deserve greater proactive attention. Even if you are working on applied areas, you need to understand this. You need to think about this, because remember, when people discovered heterochromatin, they all had their simple microscope, and but they had good thinking power. Today, with so much information, somehow our brain has got suppressed in thinking, original thinking. We need to get that back. We need to think and make use of the modern techniques and develop and understand these diversity of living systems. And therefore, biotechnology applications require integrative understanding of biological systems and their processes. And I'm sure this is something that will happen, and we will come out with real applications. So with that, I come to convey my best wishes to the Institute on its Foundation Day. And thank you all for being here. And of course, my module and the thing that led me to 93D PUF. So I wish you all a very happy future, all the young students, the faculty, that great things can happen. This was my first visit, looking at the campus, looking at the facilities. I'm sure I'm going to do it in the daytime. I'm going to enjoy it. And so my best wishes. Thank you. I mean, if the time permits and if there are questions, I'll be. Yes. There's asking.
Other important non-coding RNA. Does SSR omega gene also break down into smaller?
It doesn't break down. It produces some RNAs. We still do not know where it breaks down. It doesn't. You see, it's not that they will randomly break down. Some of them, for example, some big RNAs break down to produce microRNAs. Many microRNAs are produced as a battery of long RNA. They are produced specifically to produce, or for example, ribosomal RNA. From 45 RNA, we get 28 and 23 years and 5 RNA. So there are specific genes that do that, but not everything will be uniformly done that in that process.
So, first of all, I would like to link Form 60, 70.
Well, that's the advantage of age.
So I like to continue this. My question is that, okay, these LNC RNAs, the challenge what we are facing is that, see, they are very stage specific, like, at 2 days post-infection, 6 days post-infection.
Yes.
Any of the, like, we are
Yeah.
so like 2 days post-infection, 6 days, 10 days. So they are very, something like very stage specific.
Yes.
So if any LNC RNA which we are finding at 2 days, we will not find that LNC at 6 days,
Sure.
10 days. In case of this itself is an LNC RNA. And what we are seeing in our article in Bosworth, that okay, here we are having another some like 7 LNCR we found, but they are controlling the system, but they are again very space specific. So we are not able to
Yes.
we are sir. I mean,
Well, yes. Yeah, I agree the dilemma, but yes, but this is what we need to understand. Biology is not simple arithmetic. Biology goes in a complex manner, and that's the beauty of it. That if the same answer was produced all along, you won't get the diversity, you won't get the development. That's what is important. This does a different function. It's a long life function, and therefore it persists. It's made new, but that it's doing same function. Yes.
But again, the second LNCR, the second layer, which is
Yes.
controlling the system.
Right.
It is not
Right. So that's right. The LNCRNAs like T6 and the others, they are required only to establish this transcription and its binding with the X chromosome. Once that is done, they disappear, their job is done. Now this and its epigenetic modification that has happened can take care of itself, maintaining it. So every time cell divides, it passes on that information to the next series of cells, and so same X continues to be active. So there, you don't need that every time those the other genes required to be active. They have set the stage, they made the epigenetic modifications, which can then maintain their memory and continue. That's the interesting point to understand. So it's not that every cell, once it is born, will have to reinvent the whole embryonic two-day stage or so. No, that has been, and that's what epigenetics is. That you set in the process, it now remembers and can self-maintain itself. So in biology, self-organization and maintenance is very important. That once we have made our body, we maintain it ourselves. We don't need anybody outside to keep, unless you fall sick. So that's where I say biological systems are some of the best machines, which can go wrong for 3 reasons. One is manufacturing defect. We have genetic disorder. That's manufacturing defect. Second is use and disuse. Any machine will go bad. We age and disappear. That's use and disuse. Third is misuse. This is what's happening more and more in the modern lifestyle. And therefore, we have all these problems of obesity, diabetes, hypertension, and so on. These are misuse. And that's what we learn from biology, that it can maintain itself precisely well. And that's what the example that you are seeing is one such example of maintenance. That once this machinery has been set up, this machinery and the PRC complex that goes with it, the methylation and so on, now you don't need the other players. It can maintain itself. And that's what happens. Yes.
One of your points is that LNCRNAs can also code, I mean,
Yes.
mRNA.
Yes. Yes.
So what are the properties of, does all LNCRNAs?
No, not all. Not all.
What are the properties?
We still do not know. See, for example, the HSR omega RNA that it makes 7. One particular RNA is transcribed 1.9 kb. It is spliced, is polymerated, transported to cytoplasm, and then it has a 27 amino acid peptide which is translated. What the rest does, we do not know. What the intron does, we do not know. How the others are doing. Nuclear RNA also carry that intron, but that RNA doesn't go to cytoplasm. So each one has a different property. And you see, the important point is expect uniformity in the systems. Diversity is the basis. And so it can create more complications for us researchers or students, but that's what system is. Yes.
I have asked you were working on non-coding RNA only, but also you are working on Ayurveda and neurodegenerative disease. Suppose I'm a young researcher and I'm doing research on bacterial disease, but I also wanted to do research on cancer. But the area is very vast and lots of tower there, research is going on. So how far I can go?
As far as you want to go. Limit is only what you want to do. See, you have to read. For example, let me say my own career. I started as a classical zoologist, read whole of comparative anatomy, bone comparison between all the vertebrates, heart comparison. This was my master's degree. I didn't know DNA at that time. But then as we moved along, as I said, from parasites, I moved to genetics, learned it. Then we learned molecular biology. I didn't know biochemistry. I still don't know physics and chemistry, but learn the useful parameters of it, take help of others. Students know better, so we can learn from them. And I should say, I'm happy. Our lab was the first to create transgenic Drosophila in India in '95 with our own efforts. We didn't go abroad for training. We had conducted a workshop where we invited experts. They explained things, and then we tried ourselves. So when I moved to Ayurvedic biology in 2007, I had not learned Ayurveda at all. But yes, I had interest in this. And it was always my idea that polytene chromosomes provide a beautiful system to look at what a molecule can do in a cell, because you can see gene activity happening in a microscope. And so that's what led me to think about it and start with it. And then we began it. So if you are interested, you need to do studies. You need to do hard work. That's important. But then, see, for example, I'm sure you all have heard about Samuel Benzer, who did the R2 locus fine structure, who gave us the idea of cystone, recon, muton. It took 10 years to do that work. But prior to that work, you know what he was? He was a physics student working on transistors, which allows us the, I'm sure many of you would have seen transistors, radios, small radios, compared to the old radios which worked on valves. Now where the transistor they could be, and now we still have smaller chips. But transistors he worked, and that made the basis. But you know what happened? In Caltech, where he was doing his PhD on transistors, he once heard a lecture of Max Delbruck, another quantum physicist, who is called father of phage genetics. Max Delbruck was the one who started using phages for doing genetic studies. Why? Bacteria are very complex. Eukaryotes are forget about it. Phages with only DNA and RNA, I mean protein or RNA protein, are beautiful material to look at what the genetic material does. And so he's father of phage genetics. So he was giving a lecture, and Samuel Benzer happened to listen to it. After PhD, he forgot his transistor physics, jumped into phage, P4 bacteriophage, and he did that. And then it didn't stop there. The neurodegenerative that you mentioned, again started by Samuel Benzer. Behavioral genetics in Drosophila. He was the man in 1961 who said, now I need to move to higher organs, and picked up Drosophila. And he found out the first mutants that can control behavior. He found the first mutant that he's father of chronobiology. The Tim and Purvis were discovered in his Obed Siddiqui, who was the kind of modern biologist in India, who were trained with Samuel Benzer, partly. And so he did that chronobiology. Then when recombinant DNA technology started, Samuel Benzer made the first brain cDNA library. What all transcripts are made in brain. Having done that, he got interested in neurodegenerative disorders. In 1990s, he created first Drosophila model of Huntington's, taking the Huntington gene from humans, putting it in Drosophila, and expressing it and determining, and that's what we now keep on using. So the example that I'm trying to give you is, there's no limit. It's unfortunate Benzer never got Nobel Prize for whatever political reasons it is. But the contribution that he has in biology, beyond transistors, go very, very far. And this slide actually tells you how it is completely unrelated fields and be extremely successful. So if you have interest, all you need is hard work and brain to work equally hard, and that will do it. Only thumb rule that they don't make proteins in general. In general, not every time. Unfortunately, no. They can be polydilated, they can be spliced, they can be circular, they can be linear, they can be small, they can be large, they can be 20 kilobase, they can be 2 kilobase, nothing common. The only common is that by and large they don't make protein, but some of them do that also. So we can't have a common rule. Very puzzling, yes, but that's what it is. Challenging.
Welcome, sir. My question is very general. Like, don't you think these non-coding RNAs are reserved for next coding RNAs?
Well, they can evolve. Yes, they do evolve. Many of these non-coding RNAs, you see, this is what transposons do. That's where they have been repurposed. That lots of genes that now you see in Drosophila, we know in great detail, and because hundreds of Drosophila genomes have been sequenced in different species, so we can compare between them. And what we know is many genes have been retroposed on a new chromosome. And initially, these genes are transcribed in testes. Now, people have been wondering why in testes. Because they kind of a playground. And in the process, they sometimes acquire new functions. For example, one of the genes that we have been working at HSP60, the chaperone, that is one area that I've been working on. My lab discovered that HSP60 exists in Drosophila, but not one. But then initially one gene was discovered, then genome found out 4. And what we have now shown is that these 4 are derivatives of one single gene in X chromosome, which retrotransposed to chromosome 2 and 2-3 times and evolved, and each one has evolved. So many non-coding RNAs can start as pseudo RNA. Pseudo gene, that they are process RNA that have gone back, retroposed, have no regulator, no function, but they transcribe, and then selection makes them random mutations and repurposes them to evolve new genes. That's how we got all the immunoglobulin genes that we have. So in evolution, when you look at the duplications are provided real raw material. And many of these non-coding RNA, when they have retroposed, they have evolved new functions. Yes.
So have you tested this in other stress conditions? I'm coming from this. Do this act as stress cultures to keep proteins as a backup?
Yes, yes. See, many stresses, particularly those that disrupt oxidative phosphorylation. For example, all the heavy metals, sodium arsenide, all of them do this. The protein moves in the same way. This particular protein, omega speckles, not everything. There will be, I mean, this is not the only example. This is one example. Others can do it differently. So there are each speckle, each protein will behave differently depending upon what its function the cell is. Yes.
In my TPS or maybe 2 years more, all these signaling pathways that we that are based on our reduction control of interplay of competitive kinase and protein-protein interactions. But it's so much of involvement of all these RNA, non-coding RNA. All these steps
they will not disappear. They will be there. They will be there, but we'll learn more about it. We'll learn more. They will get more complicated. That's what life is. But we'll understand them better. We'll appreciate them better. That why same, for example, RAS pathway. Why it's involved in hundreds of different pathways. Because of context, because of cofactors, because of other signals that can happen in a given cell, not in another cell. And that's what we will begin to understand.
As you know, like here in NIT, we work for animal welfare, and as you described, like LNCs are like non-coding RNA are highly diverse and non-reproducible. And if you see a opposite side, like for flight work, we need like non-diversity in the target and highly reproducible. comment on like how we can reduce this gap to.
You can't reduce this gap. This will be there. You have to appreciate it.
Yeah, it is, sir. But
You see that this is where if I compare modern medicine with traditional medicine, say Ayurveda. Now, Ayurveda, I know there are lots of problems, myths and so on, but then the basic principle of the holistic approach, that body, physical part of body is not separate from your mental part. Physical part, one component doesn't function independent of another component. Same thing we need to learn in this process as well. That we want to modify something, we want to, through application, make some new product or something, we need to work out the total network as far as we know. And that's where, as we learn more about these networks, our approach will be more precise and more functional.
yeah, it is, for example, like you mentioned, like we can use a therapeutic process as a biomarker. So most of these biomarkers are based on the network. Okay, so maybe this is like a stage wise can be varied. So we cannot say it as a biomarker. So would you like to redefine this like
Well, you see, yes, the biomarker concept, I don't feel very comfortable.
yeah, it is.
That's again, okay, because I look at it, therefore, see, it's a dilemma sometimes. Like to some students, I'll say in class, you need to focus. You know, you need to focus like Arjun. You see only pupil of the target. But if you remain that focus always, you're lost. So it depends upon the context. You can focus, you can look at it in a wider perspective. This is a real challenge. I mean, today's biology is becoming much more challenging as we learn more. And that's what the excitement is. Okay, there was, you wanted to say something?
They are the biomarkers, but at very, I mean, very big condition.
I'll partly modify that point. Condition specific to the extent that we know today. Yes, we haven't looked at other cells, we haven't looked at other stages, we do not know, because see, the term in molecular biology use moonlighting. That a protein want to do this function, but it is also doing something else. It was at one point of time called as moonlighting. But I think that was again an ignorance. No protein is working only in one particular pathway or one particular action. You see, even the typical Krebs cycle enzymes, they can be histone modifiers, which are normal in mitochondria, but some of them are also histone modifiers. So biological system takes advantage of everything that you can take. And I can draw similarly comparison like this. You have nuts and bolts. Depends on where you want to fit it, they do different functions. But the basic thing is same. So same thing happens in biological systems as well.
Yeah, that was my question. Like, so we need to redefine like a biomarker in context of like a non-coding, because a lot of people coding like a.
Yes, there are a whole set of papers with great excitement. I've found a biomarker, but it ends there, unfortunately. See, it needs to go beyond. Whether and so you see, I read these papers. Last line says it's a good therapeutic potential, but that potential is rarely realized because they go on to hunt for another biomarker. And that's a problem. We need to move out of that area, that kind of area. Yes.
I even most of us might have realized when we studied that 1 to 2% of the DNA we all might have thought that we are studying it for the last 100 plus years, but we have been there for millions of years in our body. Doesn't know what it is doing. There's 90% of DNA whatever it shows when we feel that whatever I use the partition
Good.
So thank you for that. So another thing was for the time of the particular show. Now, RNAs or some DNA or and in the map the again for action of the body. So it's not to see how we are doing and like the time that also needs to be defined. So if we find each time
I must make one point clear. I'm not depressing anybody from following what they are doing. No, because each one does one particular thing, but then all that I'm trying to say is look beyond, look wider, have a wide angle lens rather than a tele lens focusing on a particular thing. What you're doing is what you're doing has a relevance, has actual knowledge, but then always try to think in context. I just said that you felt that 98% DNA cannot be just useless. That's a good approach, but then we need to find out what it is, and that's what we begin to look at the context.
So, following the whole cycle of this, considering other factors involved, will give us wider angle.
Sure, it will. The more players you take in, wider interactions you begin to see and begin to appreciate. Okay.
So I wish to hear your comment particularly on the long non-coding diversity. What's your comment? Why they were so diverse and how do they end up? Are we going to expect more?
Yes,
like
we are going to discover. See, no, you see, the point is, let's understand. We talk of model organisms. We study, they come out inferences from model organs. How many model organs we have studied? 10, 20. How many species are there? Billions of them. So obviously to think that all will fit in this model is wrong. So there are organs that live at boiling temperature. We can't understand their physiology. They are living at -20, -30, -40 degrees centigrade. So each one will have different diversity and different way of managing their things. They also have DNA, RNA, protein. What we learn is proteins that go beyond 50 degrees, they denature and lose. Now, how do these thermophilic and Taq polymers that we all lose every day functions? At 92 degrees, it doesn't get lost. Yes, we can think of, but then that's where we need to understand. If I say, okay, let me put Taq polymers in my body, it won't survive. It can't do its function because it needs that 72 and my body can't function 72. I mean, Taq polymer reminds me of something Harvinder Khurana stated way back in 1971. He had visualized PCR. He said, if we can have a system where we can put DNA and the RNA polymerase or DNA polymerase and precursors, and we keep on denaturing, renaturing, we'll have large more amplified. But with the caveat that we need to add polymerase every time, which when the Taq polymers are discovered, we don't need to add that, and therefore it becomes. So you see, possibilities are there, but we need to understand the context and the diversity that exists. And this is endless, absolutely endless, because all transposon elements evolve at a much faster rate than our genomes. And the more they evolve, our genomes will learn to make use of them in different ways. And so if we think that Homo sapiens has stopped evolving genetically, technologically, and academically, we are evolving at a very rapid rate. But even our genome doesn't sit quiet. We may not evolve as rapidly as other organs do because we have accelerated from environment. Hot outside, we are comfortable here. So our genome doesn't need to think of ways to combat with that situation. But still it evolves, it keeps on changing.
So I would.
Yes.
Wait.
Yeah.
Let us, okay, ma'am. Sure.
Yeah, I don't mind. Yes.
Question.
example, species that are
Frozen.
not genetically controlled and have a very wide generation gap. For example, like humans, look at what is happening at the genome or a regulatory level from Neanderthals to the current day, and what can we learn from it?
Well, I can't comment on because I have not followed that, but people are looking at human genome sequence evolution in different populations and so on. There are, and lots of studies are going on. The problem with the Neanderthal and so on, there's so limited samples that we get one sample and we make big empire out of it. Somebody makes another sample and things begin to fall flat. And so unless you're a large number, we can't really say which is Neanderthal specific sequence and which was also in Homo sapiens. Because in Homo sapiens, even the genome that we think we know, how many individuals? 1,000, 2,000, 10,000? We have billions of them. They are all not same. We talk of, you see, even the first human genome, we still don't know the identity. Probably it was Watson. And that's why I was worried that he had a Negro specific gene also in that genome. So, you know, but then the point is that one single genome cannot be ideal genome. So we talk of SNPs, we always refer to that. What is this? Because how do we say that that is standard? Not my DNA is for that particular gene is standard. So, you know, these are very relative terms and we should be conscious about this.
So, my question is like whether do they have any thought outside the cell, like
yes, they have. They have. They have very important roles. There are lots of RNAs, you know, exocytosis, exosomes are there, RNA circulating, but they have beyond that also. They have transgeneration effect as well. So what we learn typically is that all the cytoplasm comes from egg, and so we have maternal contribution, maternal dowry, and paternal contribution only nucleus. Again, wrong. Paternal contribution, even that nucleus contains lots of RNA. Outside the sperm head, within the sperm head, there can be RNAs that can be transmitted. And we now know that some of these transmitted consequence of epigenetic changes can go for 3-4 generations. This has been established, not only in Drosophila, even in mouse, in other model systems, that something that was epigenetically modified, a particular behavior modified, not genetically, but epigenetically, it can pass on over 2-3 generations. So that does happen.
So they can be secreted into the blood.
Yes, yes, it can pass to the blood, to the gametes, and therefore to next generation. This does happen.
So as you said, the nuclear DNA contributed is a wrong concept. But do we have really all that data at the level to show?
Yes, yes, there's enormous data on transgenerational epigenetic modifications. And in this, RNA plays a role. I'll give you one example, which some of you may not be familiar with. One of the parasites in horse is roundworm, Ascaris lumbricoides. And in 1890s, again, not 1990s, 1890s, Ascaris development was studied by a great biologist, Boveri, Sutton and Boveri, that name many would have heard. Boveri examined the development of this Ascaris egg, and what he found was that first cleavage, you get one lower cell and upper cell. Lower cell will undergo normal mitosis. Upper cell will now divide in this direction. And interestingly, this will lose 95% of chromatin, will not go to one of its poles. And this is precisely cut at different positions. And the lower cell will again divide horizontally. Upper cell will do that. Lower cell will remain the germ cell. So germ cells retain the entire genome, but the upper cells that keep on producing become somatic cells. They lose 95% of DNA. Now this is not random. And people have wondered, how does it happen specifically? And now we know it's because of the RNA that is passed on to the egg that determines which is required to be expressed and which is not required to be expressed as somatic cells, throw that out. And we know RNA is very role. Same thing happens in ciliates, macronucleus versus micronucleus. Macronucleus is actually a bag of genes or DNA molecules. 95% of DNA of the macronucleus is precisely cut and thrown out in macronucleus. How do they do it? Previous generations RNA that were there, they are matched. What doesn't match is thrown out. So RNA has this important role.
Mitochondrial DNA does play from the maternal side.
Yes,
only
yes.
as-
Well, that's a different story. The mitochondria being maternal is a different story. That's again why it has evolved. We do not know. Partly because paternal mitochondria are not normally passed. But there are organisms that retain only paternal mitochondria, not maternal mitochondria.
Actually, why did I pinch it? Because what we were taught and what we have been teaching is exactly this. And that's why because you did the one who may enlighten that.
Yes, yes, things are changing. Man, as Meyer has said, that much of the population genetics will have to be rewritten, much of biology needs to be rewritten. Much of molecular biology needs to be rewritten, re-understood. Yes, there was. Yes. Well, you see, practical applications can happen. Then there are certain RNAs that have very stereotyped function, and they have a specific state-specific function. You see, non-coding RNA are not random; they are again state-specific, particular self-condition specific. So one can manipulate them and do certain kinds of things. But then one has to understand what other implications can be. So it will depend upon, first thing, we need to know the full biology of that RNA, what it does, when it does, and how it can function. Then we can try to manipulate in a certain way, because you see now what is also happening is, for example, in the Drosophila we do it, we make a microRNA, we make an antisense RNA or RNAi, and we can say it will express only in this particular cell, not in other cells. Once I know the promoter that has specific expression, then I can make it target in that way. So that's one application that can be done. This is what, for example, in cancer case, people talk of that, okay, this microRNA is down-regulated, and because of which many of the oncogenes begin to express. So we can upregulate it. All that you need to do is learn how to target it to that cancer cell, not to a normal cell. Yeah, no, possible biomarker can be there, but what we need to understand is how do we target it to that particular cell where it is under or overexpressed. So the target, that's where nanobiotechnology becomes important today. But nanobiotechnology, again, what you need to make sure is that it goes only to that particular cell, not to any other cell. So, it's combination of all these things that we'll need to understand. Of course. It has to be, and that's a transdisciplinary approach. Multiple people coming together, different ideas becomes important.
One more last.
Okay.
Hello, sir. Okay, I'll be the last one then.
Okay.
No, carry on.
Okay, the student, right?
Yeah. Where? Oh, okay. Yes.
Sir, in case of X chromosome inactivation, we all know that DNA methylation is playing important role. So you told that LNC RNA is also playing role in that case. So how it is?
Well, actually, see what happens is between the two X chromosomes, there's a competition. One of them wins and becomes inactive. Now, this inactivation requires, before GIST can come in, certain other non-coding RNAs to come in. They activate this gene, and once GIST RNA is activated, it will provide platform for the methylating system, for the DNA condensation system to come and sit on the exon, and thereby methylate the GC base pairs, thereby bring in the PRC complex, the histone methylation. All these are done, and then it will inhibit the active X chromosomes GIST gene. See, it must do two things: keep itself active, but inactivate the homologous chromosomes GIST gene. So this is done, and once this has been done, so GIST RNA provides a platform on which the chromatin condensing machinery can sit and modify the chromatin.
Since epigenetic is not only regulated, it is regulated by LNCRN. This epigenetics is regulated by
Yes,
LNCRN.
epigenetic changes that happen on this inactive X chromosome are regulated by this non-coding RNA.
Thank you, sir.
Yes.
I'm going to take you away a little bit from this LNCRN. I want that maybe you can tell us a little bit more about the approaches on Ayurvedic formulation research that has been initiated and got a huge potential from countries to take it up. But at the same time, the same threat of, because Ayurveda always has worked on formulation and there is a huge urge to segregate the molecules and try to understand. And in most of the cases in isolation, these molecules do not work, as we know. At the same time, Ayurveda also always has documented the impact on the whole physiology of an organism rather than looking at a very segregated either cell or tissue. And it has always worked in the context of multicellular compositions and various kinds of parameters that not only one cell contributes, but many other cells contribute. So in that scenario, when many of the approaches are that people come down again to cell culture-based assays or separating the molecules and trying to document a lot many effects of such Ayurvedic formulation. What kind of caution you will ask, and to what extent we should still continue to do that, because
All right.
it does add a lot of
Yes.
information.
Yes,
for sure.
that's a very important question. If I can go a little in history part of it, modern medicine is derived from Ayurvedic-like considerations. See, in 17th, 16th, 17th centuries, lots of Portuguese and others came to India searching for these spices and the medicinal value that they have. One such Portuguese, for example, spent about 26 years in Goa region, collecting information about every herb that was used in the traditional medicine there. He prepared 26 volumes of literature, which is available today, sent it to Europe, back, and then they began to look at it. And then at the, so initially all the drugs were derived from plant system. Even till 1950, more than 80% of modern medicine were from animal and plant origin, and they were as chemists. But then as chemical industry evolved, they began to synthesize. And our modern molecular biologists began to take active ingredients, active principles of these Ayurvedic formulations. And which is where I think things went wrong. Because they lost the holistic approach to the same molecular biology approach as a one molecule, it will. So we have all the docking experiments that I determine the structure of protein molecule and enzyme, and I determine the pocket in which the small molecule can fit in and inactivate it or activate it. And enormous work has gone on based on these. Not that this is invalid, but what the forgotten is that the same molecule, same protein, will in same cell or different cells have same structure. It may have different structures, and therefore binding can be different. And so this is one part where we need to take holistic approach. The Ayurveda principle itself is a remarkable example of huge observations. And of course, Charak or whosoever gave it 3,000, 4,000 years ago, the whole idea, what experiments we did, we will never know. Whether they did any experiments on animals or they did, but Charak Samhita, for example, describes in great detail quantity of each component, how to process it, how to add it, how to do this. We will never know how did they do it. But what's happening today is tragic. Even you take Charak Samhita, follower, somebody who says I follow Charak Samhita from Kerala, from mid of country, or from Hyderabad, or from UP, or from West Bengal, they will have different opinions. Same drug made in different ways, and they will all claim that this is Charak definition, and that's the unfortunate part. What we are also missing is, when Ayurveda was written at that time, they had a different concept of biology. They didn't know biology as we know today. They knew only from what they could observe, what their five senses could perceive. They talked of Pancha Mahabhut, each sense determining heat or determining taste or this thing, and so. And then three, what you cannot have what with the cup that they described, what you can see directly and correlate with air or heat or something. These are empirical. And of that time, remarkable. But in today's times, we keep on following it, we're in serious problem. And which is where Ayurveda is in serious problem. Because I know how difficult times I had, and I feel community. They think I'm an outlier. I should do my science, forget about Ayurveda. This is what somebody had commented. And in fact, Ayush Ministry in 2018 brought out a circular, an advisory to not only research community, but also even to journals, that non-Ayurvedic people should not conduct research in Ayurveda. Yes, unless they are associated with an Ayurvedic specialist, because otherwise they're being disrepute to Ayurveda. This is what the language was. And an advice to journal editors, do not accept a paper from Ayurveda community on Ayurveda if there is non-Ayurveda person not associated. We wrote a strong rebuttal to this in current science. Fortunately, 2 Ayurveda people joined with me and we wrote it. But that's fact is today also it continues. You see, for example, I mean, this is where I feel very sad. Professor Vaidyanathan and Chidambaram's effort, 2 non-Ayurveda people. Chidambaram was the nuclear physicist who was involved in the Pokhran blast and so on. He was principal scientific advisor. They came together and floated Ayurvedic biology. And ultimately it became a task force of SCRB, which was funding our projects. Sometime in 2016 or 17, Ayush Ministry stopped this Ayurvedic biology task force in SCRB. Who are you to fund Ayurvedic biology? And so that whole research is going on. I actually, 2 weeks back, wrote an email to Ayush Secretary that unless we go with the syndrome that we knew all, will remain only we knew. What we know is more important. Not what our great forefathers knew. It's very important. It's very important. I mean, I've been writing about this. For example, recently I wrote to a small letter to one of the editors that we need to get rid of myths in Ayurveda. We need to get rid of, okay, Vaspita we talk, but this is the personal medicine system, right? This is what has been touted, that Ayurveda only provides this personal medicine system. Talk to 2 Ayurvedic people, same person, they'll classify differently. Fact. It's not an exaggeration. Because the definitions are so empirical, walks very fast. Well, a fat man can also walk very fast. Not necessarily a lean man. A lean man can walk very slow. So just by these simple, there have been some studies on the genomic correlates, but they've been very fragmentary. Because these studies have been done to establish that okay, what phenotype is associated with a certain allele of a given gene. Very good, but that's a correlation. It doesn't establish, it doesn't become a parameter by which an Ayurveda specialist can identify and define a treatment. But the treatment says very clear that if this is the symptom, you treat it like this. You see, even Amla, Charak has studied very carefully, very clearly, that Amla grown in different seasons. Amla grown in different parts of the country will have different values. Do we care about it? We all buy Chyawanprash, right? We don't know what it is. It's a fact that the ingredients of Chyawanprash, they are listed. If the quantity of Chyawanprash that's made in India, we don't have that many herbs in the country. No way. And I can tell you, we tested it in Drosophila. Some brands, I will not name them, 50% have killed my flies. So please be careful about it. Amla can be a wonderful thing. It is. Certainly it is. But then, depending upon how it is processed, how it is done, what else it contains. I've been discussing with a foreign group, a group based in America, who want to, promote Ayurveda. And there were a lot of industry from India. And we had a group discussion, it was 3-4 months back, that why are Ayurvedic formulations banned in US and Europe if they can help there and promote it, so much of money will be earned and so on. I asked them a question. You are worried about your quality control for US product. Why not about Indian product? Why don't we apply the same criteria that our product which is sold in India must also have quality control, which we don't have at all? Because that's what I said, that if Chyawanprash, which is so widely used off the counter, almost everybody takes it. I'm sure even here, more than 50% will are using Chyawanprash. It's a good regenerative system, provided it is made properly. If it is not, it can be dangerous. And they are very heavy metals. Heavy metals, that's why US brings it. We have also shown that Rasindur, which is pure mercury sulfide, it's completely non-toxic. But if you take its intermediate product, it's completely toxic. So unless you process it in the manner that does, and that's remarkable that 2000 years ago they discovered this. And I was reading how they decide how much to grind the Rasindur. Something very interesting, very empirical way. They make nanoparticles. I'm sure they didn't have anything to identify nanoparticles. They didn't have a kind of microscope that can tell you whether it's so many angstroms. A simple definition was take a clean water in a tray of water, white color tray, sprinkle a pinch of it. If you don't see red color, it's well grounded. That's it. Because it is so small that your eyes can't see. Now, these are empirical, but today we need to do it better, and that's what I think Ayurvedic biology wanted to do it, provided it is allowed to continue that way.
It will still require a lot of organic level.
Yes.
But to what extent we can still take it to cell?
You see, cell culture can only, you see again, yeah, cell culture, organoid, and so on, it provides us some leads to begin with. Then, or we can take, for example, what we found in Drosophila may not apply directly to humans, but it gives us a lead that yes, these can suppress typical neurodegeneration, Huntington's or Alzheimer's. It can then of course with a collaborator in CCMB, which is in mouse, the same thing happens. Then one of my collaborators in IIT Kanpur, he works on a Lafora epileptic disease. And there again, Rasindur does, and Amlodipine does wonderful things. They also found it. But then for treating it with humans, I think Ayurvedic people need to do some studies. Instead of simply taking it, okay, because yeah, they take advantage of this, but they don't want to do the hard work themselves. They don't want to come out of their shell. I mean, I talked to several of senior people. They say, Lakhotia, don't talk about Panch Mahabhut and all this. That's not your domain. I can't help it.
Thank you very much.
Thank you. Thank you.
So we thank sir for a very informative and interesting talk. As we can see that it was full of curiosity from the students as well as from the faculty, and the discussion happened over here. So now with this, I would like to invite Subir sir to come on the stage and address the audience.
Thanks, Sonu. And it's a great occasion to have Professor Lakhotia here. And one of the things which Sonu didn't say, that he is an excellent, extraordinary teacher. And before I forget, I should say thank you very much for coming to us, sir, and see our institute. It's a good luck for us. And the number of questions which came today is much, much more higher than we have seen in the past. The reason is very simple. The way he delivered his talk actually went into the zone of understanding of each and everyone sitting here, and they could relate themselves. And I'm worried, sir, that after this, everybody would start working on long non-coding RNAs. And their guides will have problem in convincing them. So it's very interesting. And sir's HSR Omega work is very famous. I have known sir through INSA, and he is a great writer. And if you get time, just go. He was into science and policy writing also. He was twice vice president of INSA, before that council member. And he was also editor of proceedings of INSA. And he also wrote a very good document. If you get time, you can read a policy document on science dissemination and evaluation on research output. So some of these things, since you asked, he told you that he writes, but you can go and see. That's a great thing, because in addition to being a good scientist, when we all grow up, it is our responsibility to modulate the system in a way so that the research is favored. Sometimes, as he was saying about the Ayurveda, we do not get favorable environment to grow ourselves in a certain field. There are difficulties. Policymakers or maybe inter-scientific interactions, everything. But if that is brought up in a platform and discussed, life of people following us becomes good. So that's a great contribution of him to science and society. I'm really grateful. He was also executive council member of International Union for Biological Sciences, and then also vice president of that. So he has great contributions to Indian science. He is known as a very serious person. And I mean, simple thing is, when he got Bhatnagar Award, I submitted my thesis. I got award of thesis on that year, 1989 to be precise. And he's respected all around. And if you consider that if you go into the fields of cricket, people used to say, Sachin will retire the day he wants to retire. Kapil will retire, he wants the day he wants. Dhoni will retire the day. But that doesn't happen in the field of cricket. But that does happen in the field of science. So if you take few peoples I will name, you can correlate. One of them, he himself said, Professor Valiathan, professor in biological sciences, I am saying, Professor Balram, Professor Padmanabhan, and one of them is Professor Lakhotia. The day they want, now I finished my batting, I don't want to bat. Otherwise, they are very much respected in the field, very much in demand from people. And they are such a mature brain, such a learned brain that everybody would like to interact and give some information, knowledge, idea, guidance. So we are very lucky that he was amongst us. And long non-coding RNA is really a very interesting subject, and a lot is yet to come. As I think Prasad asked the question, I think we don't know what we are going to see. But my last PhD thesis, my student submitted from NI, was on kind of negating long non-coding RNA. Two of them she did. One she didn't get any information, but other she got information which is so difficult for me to correlate with my science that I said, okay, now you submit thesis, we'll think about paper later on, because it really disturbed a lot of things. And I personally think our colleague Souvik has started working on endocrine disruptors now, and his interest does lie in the epigenetic regulations. And while writing that project, I was kind of helping him. So I went to the literature, and it appeared that from nutrition, which is a kind of Ayurveda, you can reverse the epigenetic regulation. So now you understand epigenetic regulation has to occur in the nucleus. So probably there is a great role that some, maybe with the Ayurveda or the nutritions and the feed we take, we can generate either new non-coding RNAs, or I don't know, suppress something that epigenetic changes can be reverted back. So I think it's a great scope, it's a great field, and he has given us a very good idea. And youngsters can, so that is the benefit to all of you, that you can think that way. And I remain grateful to sir for coming here and giving us so much of time, love, affection, and interaction. He enjoyed. I think that is very, very important, because he is a so famous teacher. The teacher would always enjoy if there is a great interaction. And I am thankful to the audience for being so curious to his subject and really interact so well with him. And I wish sir a great long innings and retire the day really he wants to retire. Thank you. Thanks.
Good afternoon all. This is time for prize distribution, as all of us gathered here for 11th day foundation day of NIV. So I request Professor Lakhotia sir along with Dr. Hegde sir on the dais. Please, sir. So with the on the foundation day, we had a different sports activity as well as some arts related activities. So we will start with the walkathon, which is conducted on 11th and 17th of this month. So with the women's team for the walkathon on 11th, Divya got the first prize. Second is Meenakshi. Third is Komal. Divya, first prize. Meenakshi, second. Third, Komal. In men's walkathon, first prize goes to Shashikanta. Second prize is to Pratik. Third is Subhashis. On 17th also we had a walkathon competition. With the women's, third prize goes to Ria. Second prize is to Satrupa. And first prize to Richa. Same day with the men's competition, third prize goes to Naveen. Second, Venkatesh. And first prize to Varad. Along with the foundation day, we had a Swachhata Pakhwada. For that also we had some sporting activity, especially Swachhata Dod. For that, women's team, their owner, there are 5 people we have selected. The 5th place is Deepa Lakshmi. 4th one is Aradhna. Third, Sakshi Singh. Second, Divya. First prize to Deepali Rawat. For Swachhata Dod, main participant, fifth prize to Rafiqul Islam. Fourth prize to Venkatesh. Venkatesh Security. Third prize to Harish, Engineering Section. Second prize to Subhajit Mandal. First prize Shashikant. Thank you, sir. Thank you so much. I will call Dr. Subir Muzumdar, sir, to come on dais for the next session of prize distribution. So we had a different competition, indoor competition as well as outdoor competitions. So we'll start with the Carom women's double team. The second prize goes to Ria and Ritvi. Carom. Double. The first prize goes to Devasmita and Nilam. Carom's double. Men's double, second prize to K. Vinod and Azhar. First prize to Gopal and Mithun. For chess competition, second prize goes to Suresh. First prize, Jivan Ratnam. For chess, second prize, Suresh. For table tennis, women's double, we have runner-up Priya and Binita. Yes. Women's double, second Runner-up Priya and Vinita. Winner is Akanksha and Devasmita. Men's doubles, runner-up Varad Deepak. Winner is Vivek and Anchita. Achinta. We'll move on to next tournament, that is thank you, sir. Thank you for your. I called Director Ma'am on dais for distributing the next prizes. For badminton women's double team, runner-up is Akanksha and Devasmita. Women's double winner is Aradhna and Anjali. In men's double, runner-up Anuj and Sanjeev.
On the WhatsApp also. Screening not
Men's double, runner-up Anuj and Sanjeev for engineering section. The winner is Venkateswarulu and Ramesh. Winner of badminton. We have now prize for the cricket women's team, women's cricket matches. Runner-up is Patricia the Scarlet.
Beside ground. Besides starting.
One showroom, car showroom, opposite the Nexus showroom.
Very nice.
In men's cricket, we have runner-up team students. Students is led by Dr. Gaurav Kumar. Team, students team, come on guys. Men's cricket team, young star from admin. Sayad. So there are 11 entries came for this competition. Out of 11, we have selected the top 3. So third prize goes to the volcanic intuition from Dr. Panula.
No, not this one. इसके अलावा this is the
second prize goes to Moonlight Flamingo from Dr. Ramesh. And first prize goes to the butterfly effect from Dr. Kalishna.
बाद में आना है
Thank you. I will call Dr. Paresh for a vote of thanks.
Good afternoon. So everything good thing comes to an end, and today I feel incredibly honored and privileged to be allowed to extend a vote of thanks on this auspicious occasion on behalf of NIB. I propose my most sincere thanks to the guest of honor, Professor Lakhutia, for taking time from his busy schedule and gracing the occasion with his warm presence. It's indeed a great honor for NIB to have welcomed such prominent speaker, known for his great contribution to their respective research field. I am sure we will greatly benefit from his opinions and findings today. And this NIB Foundation Day, or as we are celebrating NIB Foundation Month, was not a one-day effort, but many individuals have given their hearts and soul to make this foundation event a success. Of course, behind the success of every program lies the effort of sincere, experienced, and talented people. And we cannot take this credit away from our seniors who have constantly inspired us to excel in whatever we do, whether it's work or any other activity. I would also like to thank our director, madam, on behalf of the whole NIB for promoting such activities and giving us a free hand in carrying them at NIB. Enormous cooperation was received from the administration, engineering, security, and housekeeping staff in arranging the event, ensuring the availability of various facilities. We are also quite fortunate to have worked with lots of dedicated and sincere students of NIB who have made themselves available whenever required and helped make this event possible. I now conclude my remarks by wishing everyone prosperity and good health. Thank you very much.